Okay. So here we are again for our learning on transgenic animals and how exactly do we produce transgenic animals. And we were almost at the end of that particular session, which is talking about, just give me 1 second, okay? Okay. So we were having this application part we learned about. So yes. So I hope we have means like we have learned many things by far in transgenic animals, like how exactly you want to go about the process of producing transgenic animals and what all importance and what all applications it might have. That is what we are talking about. So industry importance we have medical importance, we have agricultural importance as well, in which we can produce animals who are disease resistant. We can also produce plants who are disease resistant. Since we are talking only about transgenic animals, I we are explaining only about animals, but we do produce transgenic plants as well, where we exhibit the same properties, the type of properties we want a plant to have, and we want a plant to be disease resistant. We want them to bacteria resistant or some disease fungus resistant. So all that properties you can inculcate into plant or animals using this transgenic models or something like that. So for improving quality quantity of milk, meat, eggs, and wool production, for that purpose also we can use this transgenic animals in the agricultural site of our studies. Then application, production of an organs for transplant, that is also what we can do, right? By changing a stem cell, by introducing genes into the stem cells and allowing that stem cell to grow, because we all know that stem cells are the one who get produced, are the one who get multiply and can produce an organ. So when we are saying an embryo is nothing but a stem cell which can further grow and mm, form into an organ or some part of our body. So that is what also can be done using stem cell technology. Shraddha, you are there for the lecture? Is Shraddha around? I can't see her here. Join the lecture though. Mm, just give me. I have this message to be taken from the admin. Uh, okay. So, yeah. So, application part, production of organs for transplant, surgical techniques, cancer models, antibody production, and study of a role gene. Study of a role gene is the same thing that I have told you, that you might delete a gene and then you will see what kind of animal is getting produced. You might introduce a gene, then you can see what kind of animal is getting produced. So that's the trial and error and research scope for many scientists they had before, and that is when we got to know what are the lethal mutations and what are the okay means like not so effective mutations. It's like even if you delete that region, nothing happens in the newborn baby, right? And some of the deletions could be such that it might lead to some deformities in the body of a baby or something like that. So that is what you can do. And we are going to see this particular antibody production part in detail through hybridoma technology. What could be the issues related to this transgenic technology? So there may be health risks associated with the transgenics, right? There may be long-term effect on the environment when transgenic animals are released into the field, right? Abnormalities suffers are more, reduced fertility, respiratory and circulatory problems can be there, and weak immune system, that could be also one of them. So, what would be the conclusion of the whole thing that we have learned by far? That with proper research and careful use of transgenic animals, we can go a long way in solving several problems for which science doesn't have a solution till now. So, manipulating your gene and making some changes or making some changes that can lead to good property of the zygote or good property of a person where we can save that person from any kind of a genetic disease or to be held genetic diseases. That is what we can achieve by using this transgenic animal models, and we can check at various time points, like if I do this, what will happen? If I perform this kind of a mutation, what will happen? If I introduce this kind of a gene, what will happen? And that is possible only through the way of trying it on animal first and then implementing on human beings. So if you make a cluster of whatever we have learned by far, so it starts with DNA cloning and ends with making a transgenic animal, and then having this transgenic animal model getting implemented into human beings. Okay, for this also we have clinical trials. So you see, there is a whole circle. It starts from in vivo. So हमने जो clinical trials देखा था, that maybe we talk with respect to only what you say. Medicines or something like that. But if we do some models like this, if we implement these models in human beings, then also we have to do go through the same kind of procedures as that of a normal clinical trial for a medicine, right? So it started with learning about restriction enzymes, then it started with learning about what is DNA sequencing, what are restriction enzymes, and what are clones, what are vectors, like what are different vectors, and how we can use cloned DNA into this particular making of transgenic animals, and how we can further go and use this. If it is successful, how we will be going ahead with the clinical trials. That is when we will be going ahead with the clinical trials. So I hope I have made it clear what exactly is what this transgenic animal means. And I have one more thing to show you, that over here in transgenic animals is, so it talks about the same thing that we have learned about. The one thing that I wanted to, oh, I'll be uploading both the slides on the Google Classroom, so don't worry about that. So one thing I wanted to tell you guys over here is transgenic animals में knockouts. Okay, the knockouts. So a special case of transgenic mice that have been genetically engineered so that a specific gene is disrupted. So that is when you will be calling those mice as knockouts. Okay. Oh, so DNA is modified and added to embryonic stem cells where it recombines with the existing gene on a chromosome and called as homologous recombination. Okay, and modified embryonic stem cells are introduced into normal embryo, and then embryo is implanted into a foster mother. Okay, so that is how the production of knockout mice is done. So the mouse pups is a chimera, and some, that means, what do you mean by chimera? I have already told you. That means some cells are normal, some cells are knockout, and two generations of breeding is required to produce a complete knockout, means until you get that homozygous knockout. Okay, so you need to breed the mice two times to get the complete knockout mouse. So knockout knock-in animals have a human gene inserted uh, to replace their own counterpart. Okay, so you will be inserting. So knockout है जहाँ पे हम जीन डिस्टर्ब करते हैं, और नॉक इन है जहाँ पे हम इंसर्ट करते हैं जीन. ठीक है. So it has same procedure as that of embryonic stem cells method of introducing a transgene into animals and then producing a transgenic animal. So that is the same way. That is what they have explained over here. So this is one more thing of knockout animals, and this is a very nice diagrammatic representation of how exactly this insertion of the gene into the mice will be done, and what all products we can get, and how exactly will be salting this uh, uh, transformed gene. Okay, I will show you, just a minute. I will explain you this. Okay, so I hope you guys can see it clearly. So we have a insert gene with antibiotic marker into plasmid carrying marker gene. So now I'm hoping that you know that what do you mean by antibiotic marker with that, right? Then you have neomycin resistant gene, that is your selectable marker over here. Then you have second part you have is collect the embryonic stem cell from the mouse. Okay, so you have insert, ठीक है, ये वाला तुम्हारा पूरा प्लाज्मिड है with your resistant gene and your target gene. Okay, then incorporate the plasmid by incubating the stem cell. So you have taken the stem cells, so these are your stem cells. Okay, just a minute. Okay, somebody's mic is on. Okay, so you have embryonic cells, and you have this particular plasmid, this vector of ours, which is having our gene of interest. Then you will be incorporate the plasmid by incubating the cells. So you basically are going to incorporate this into the embryonic cells. Then three kinds of products results from the incorporation of the plasmid, and one of this is the cell with the interrupted gene, that is your knockout. So when you're doing this, you're inserting the plasmid into the stem cells. Not all the cells are going to incorporate this plasmid of interest, right? So that is when you will be getting three products. Okay, what are these three products? We'll see. So you have homologous recombination, which will be giving cell with the interrupted gene. So this is something you want. So this is the correct combination that we have got. So you have homologous recombination happening between this target gene and the mouse chromosome. Okay. So where exactly the recombination is happening in the mouse at his chromosome level, because we are using what, embryonic stem cells, right? And then mouse gene with the inserted gene. So you have mouse gene with the inserted gene, and then you will be getting a chromosome where the cell with the interrupted gene is present. Okay. Then non-specific recombination, where the random insertion will happen, and then you will get the cell with the random insertion. Okay, so you are you this is something you don't want. Okay, then non-recombination, that means nothing has happened, cell with no insertion. So these are the three kinds. So this is something, this is also something we don't want. So these are the three combinations that we might get after combining this plasmid with the embryonic stem cells, right? So then after that you will have to do the selective picking for the one which you want, right? So you will be selectively picking up only this cell with interrupted genes. Okay, because since we are talking about the knockout model, so we are saying that cells with the interrupted gene. So you collect the cells, select with neomycin TK substrate. So now you know that my target gene, okay. See, in knockout, what is there, you know, that you will be knocking out the gene, okay, and you will be replacing that gene with something else, okay. So hence, when we are inserting this target gene, we know that in our plasmid we have added neomycin resistance, right? So select with neomycin and TK substrate. Only the cells that have the interrupted gene will grow in the medium and resist the drug that kills the cells with the functional TK gene, right? So since we have neomycin resistant over here, so selectively we'll be picking only these cells, only these ones. Okay, and then let me see if there is something. further to this. Let me clear all the drawings. I have made a mess over here. So yeah, that's it. So you have so when they are showing chromosomes over here in this part, that means some of the mouses that you're going to get will be having the chromosomes with which you can means like this cells you can then select and insert back into the foster mother of of a mouse, right? So these are the cells you are going to select, okay, and these cells you are going to put in the embryo of the mouse, and then you are going to put that embryo back into the mouse, the foster mouse. Then that mouse will give birth to the babies, and then the babies will be chimera. That means they will have a stem cells or normal stem cells, which will be having this trans target gene, and then you'll be breeding them for 2 generations, and then you'll be getting homozygous transgene, means the gene interrupted gene or knockout mouse, right? So I hope you guys have understood. Have you guys have got it by far? Are we clear still here?
Yes, ma'am.
Okay, thank you, Pratibha. Or Sheetal, somebody said yes, ma'am. Just give me 1 minute. I have to make this call, so I'll just hold the recording for 1 minute, okay? Okay. So hi. Is Tanuja there? Tanuja or Samruddhi? You're there? Tanuja, you're there? Yes. I need your friend Shraddha's number, alternate number if you have any. I need to speak to her immediately. So if you have any alternate number of hers, please
Yes,
send it to me. Okay.
yes, ma'am. Actually, she have a light issue. So
She
for
has
her,
what?
ma'am. Hello.
She has
Yes,
what?
ma'am. I can share. Light issue.
Uh, your voice is breaking. You can type it here. Okay. You can just type it here. Okay. So this was it from my side with the transgenic animals, and one limitation, let's see. Okay, so this is the part of production of monoclonal antibodies, in which also we use models for making the monoclonal antibodies. So that also we will see in detail right now, so which involves the hybridoma technology. And what exactly is the background, we'll see. So basically production of human antibodies in animals is the main thing that we are going to learn. So production of monoclonal antibody is called as MAPS. So mouse or rat inoculated with an antigen to which a antibody to which an antibody is desired. Okay, so you'll be simply, if I tell you, you will be injecting an antigen into the mouse body or the rat body, and they will be producing the antibody in their body system. So spleen harvested after an immune response is produced. So you'll be removing their spleen, and you'll be harvesting that spleen, because the spleen is the major part where most of the antibodies are being produced. Spleen cells are fused with a specialized myeloma cells, a cell line that no longer produces an antibody of its own. Okay? So you will be making, so myeloma is an antibody secreting tumor. So basically you will be taking the spleen after injecting the antigen, you will be taking the spleen cells, you will be fusing them with the myeloma cells. What are these myeloma cells? They are nothing but they are antibody secreting tumor cells. So tumor part, the tumor, the what you say, tumor part will be silenced in that particular thing. So only thing they will do, the myeloma cells will do, is produce antibody. Okay. So spleen cells are fused with a specialized myeloma cell line that no longer produces an antibody of its own. Okay, so they will no longer produce the antibody of its own, but they will only the spleen cells will produce the antibody of its own. So why we have to do that, we'll all see it later in the another PowerPoint presentation of ours. So production of monoclonal antibodies, the resulting hybridoma, that is fused cell, that is your spleen cells with the myeloma cell line, grows continuous and rapidly like the tumor. So why you have chosen these myeloma cell lines? Because you want these spleen cells to grow faster. After you fuse this spleen cells with the myeloma cells, you will be having the characteristic of that tumor, which will be growing faster. So without producing any antibodies. So spleen cells will be producing the antibodies, and at the same time, they will be growing faster also. So these few cells will be producing antibodies and they will be growing faster as well. So rapidly like a tumor and produces the antibody specified by the spleen cells. So that is called as hybridoma. This fused cell is called as hybridoma. So MABs are used to treat cancer, heart disease, and transplant rejection. So this is what the main use of monoclonal antibodies is done. And nowadays we know that monoclonal antibodies are the major technology that we need for any kind of a disease, right? Most of the times for our research purpose also we want to make some monoclonal antibodies which will be uh, helping us to understand the characteristic of the protein, characteristic of the gene, because you remember that we were talking about labeling of the DNA probes with the antibodies. So at that time you need antibodies which are very specific to some particular nucleotide sequence. So that is when you will be needing those particular antibodies, those monoclonal antibodies which are pertaining to that particular gene or that particular protein itself. Okay, so moving further, so this is the diagrammatic representation of producing antibodies in the animals. So a very simple thing that we'll just go through it and then we can talk in detail about it. So you have will be injecting the purified antigen into the mice. Okay, then you have myeloma cells. So you have put the injection, you have myeloma cells. So these myeloma cells are what? They are tumor cells, but they are not producing any antibodies. Okay. Then you will be isolating the spleen from the mice. Okay. Then you will be fusing this spleen cells and the myeloma cells, and you will be getting this hybridoma cells. What are these? Select hybridoma cells in a selective medium for marker gene. So you see, this is where main thing is. So once I fuse this spleen cells with the myeloma cells, again I'll have the combination of the cells. I will have when I try to fuse these cells with the spleen cells with the myeloma cells. So either I'll have hybridomas. Hybridomas means the cells I desire, right? They will have both the characteristics. They will have myeloma and isolated spleen cells. That is hybridoma cell. That is fused cell. Then I will have only myeloma cells. I will have only spleen cells. So what I have to do, I have to selectively select only these cells, these ones. These are the ones I desire. Okay. So select hybridoma cells in selective medium for marker gene. So हमें अभी पता है selective marker genes क्या रहते हैं, how to select exactly only the cells you desire. So over here there is one media that is made specially to select this cells, and there are some changes that are being made in the myeloma cells where which can help us for the selective selection of this hybridoma cells only. So there is one media called HAT media. In that HAT media, only the hybridomas will be surviving. All other cells will be dead. Okay. So selective hybridoma cells in selective media for marker gene seed individual cells into the wells. So you'll be putting them into the 96 well plate, each individual cell, allowing them to multiply. Antibodies are secreted into the culture media, because since these cells, hybridomas, have myeloma in them, what will be happening? They will be multiplying at the rate the tumor multiplies, because we all know that tumor has a very high capacity of multiplying than compared to a normal cell. So in each individual, lot of cells will be multiplying themselves and antibodies will be secreted accordingly. Then test hybridoma culture medium for monoclonal antibody that react with the antigen. propagate positive clones. So you'll be taking only those hybridomas who are able to make this monoclonal antibodies, and you will be propagating those positive clones only. Then freeze away a cell stock. So you'll be, since you have selected only those hybridoma which are able to produce the monoclonal antibody that you desire, then you will be saving some stock for your future use as well, and you will be extracting the antibodies as well from those cells. So this is a very simple diagrammatic representation of how exactly the monoclonal antibodies are produced. ठीक है, पॉली मोनोक्लोनल अगर कर सकते हैं तो polyclonal is not a difficult task. Okay, so this is how the production of monoclonal antibody in short is done. So my mouse hybridomas cause HAMA response, human anti-mouse antibody response, working to solve this problem by creating antibodies in different organisms. So this was it with the myeloma and what you say, production of antibodies, monoclonal antibodies. But we are going to see this hybridoma technology in detail. We have 5 minutes. We'll just go through the history for Tuxet and probably then we can stop. So in 1975, this technology developed by George J.K. Kohler and Cesar Milstein, and in 1984, they shared a Nobel Prize for this discovery. They make a hybrid cell that will make a number of monoclonal antibody against an antigen. So this is also a major task. You have to know this, that antigen specific antibody ही बनाई हुई हमें चाहिए, right? So it's such a molecular level research that you will be injecting one antigen of your of that you desire, and then you will be isolating the antibodies that are produced against that antigen from the body of a mouse. So it sounds very simple when I say it in a very simplified manner, but then there is a lot of technology and lot of research that has been involved in order to carry this experiments. Okay, so what is the principle of this hybridoma technology? The hybrid cells has the capacity of antibody production delivered from the B cell, that is B cells. That is delivered from B cells, that is your spleen cells. At the same time, it can divide continuously by the quality derived from the myeloma cells. So that is what I have told you. So you'll be having B cells from the spleen, which will have a great capacity in the antibody production, and then you will be having myeloma cells who has the property of dividing continuously, right? By combining the desired qualities of both the cells, the technology ensures large antibody production of a single specificity. Specific hybridomas, that is spleen cells and myeloma cells, obtain monoclonal antibodies in artificial media. This technology is called as hybridoma technology. So you'll be adding a selective media in order to select this fused cell, this hybridoma cells, and then taking using these cells for production of our antibodies against that particular antigen. Okay, so this is what they have shown, whatever diagrammatic representation that I had shown you just 2 minutes back. That is what is shown in a different diagrammatic presentation over here. So antigen into the mouse, you'll be extracting the spleen cells, you'll be taking the myeloma cells, fusing them together and making a hybridoma. Then this hybridoma will produce what? Antibodies. And then uh, cancerous plasma cells is nothing but your myeloma. Okay, and then you have a hybridoma, and which will produce the antibodies of our desire. Okay, are there any doubts in this session? There are only 15 students today. Why this is so? Why people are not joining the classes? If there are any issues, I have time and again telling you that you have to let us know what is the issue from your side, because in a class of 22 only 15 students are there. That means 7 students are not able to attend the class today. And we have to know the reason, and accordingly the action will be taken. Okay. So when the time will be there, when we are taking any action against the lectures you couldn't attend, that time there is no use of explaining yourself. Okay. So yeah. Yeah, thank you so much, Sheetal, for letting me know that there are no doubts. And I think we are going in a very simplified manner. Thank you, Naina, as well. And it's not very difficult to understand. You just have to have little bit of a background knowledge of molecular biology to understand this. Okay. So we are means I'm signing off for now, and we'll see you tomorrow. Tomorrow at the same time 11:30, and we'll go further with the hybridoma technology in detail. Right? What all steps are involved, and how do we exactly go about it? Okay, everyone, take care, and we'll see you tomorrow.